Okay, is a transcription is a important process, and for that transcription these factors are essential, and without this transcription we cannot have the proteins. So we know what transcription is. We shall be again, if possible, we shall have one view on this. So what are these? Let us see what are terms which are usually used. We have seen gene expression many a times, that is by which the information from a gene is used for the synthesis of functional gene product. Now it can be protein or it can be DNA itself. So for gene expression usually we have two things, that is gene is expressed and it gives some product, that is, if it is a replication it can be new DNA itself, or it can be RNA, that and which is used for a protein. So it is the expression which is resulting into this protein. Then we know what is transcription, that is it makes the messenger RNA from DNA by using RNA polymerase enzyme. Then next is the transcription factors. These are the proteins that binds to the DNA and they will regulate the gene expression by promoting or suppressing the transcription. Okay. Now these are many times we have used word repressors, or we have used the words that is that is promoters or suppressors. So this means these are the transcription factors which are acting as a promoters, means which promotes the gene expression or which promotes the transcription, and there are others which are called as a suppressor, which suppresses the transcription. Then transcriptional regulation is the term that means that it controls the rate of gene transcription for by helping or hindering the RNA polymerase binding to DNA. So they are regulating, they can increase or decrease in the rate. Regulators means they can increase the RNA formation, rate of RNA formation, or decrease the rate of. Upregulation, this term you might have heard many a times earlier also. Upregulation means increase in that particular activity, okay, or activation or promotion. These three terms are used synonymously. Actually these are slight differences are there, okay. And down regulation is a repression or suppression, that means decrease in the rate of gene transcription or stoppage of it. It can be, so it can be repression. Co-activators, these are the other proteins which will be there that will be causing the simultaneous activations. Then the down regulations, they will be causing the repression. Co-activator, co-repressors, they will helping in the repression both, okay. They will increase the repression or suppression or down regulation, whatsoever it may be. And response element, it is a part of DNA, is a specific sequence of DNA that will be acting as a transcription factor, and that will bind to the DNA and accordingly it will be there. So these are various terms which are used. This is the glossary, or these are the usually used term. Now there are variety of transcription factors which are playing important role. They are playing important role in many places because they are doing in a very coordinated fashion. They are doing in a cell division, cell growth, cell death, throughout life, cell migration, and various activities. So they are there, and therefore these transcription factors are important. Now quite a few times I have used the term that is human genome project in the beginning of this century 2000. That is during that a very ambitious and very big project was carried out, that was known as a human genome project. And it was during that it was found that there are around 1600 transcription factors are present in the human, or that human genomes can express, or human genome can code for 1600 transcription factors. So these these transcription factors, they can work alone or they will work with other proteins. They may work singularly, so no complex, but if they are working with other proteins, they may work in a variety of ways. They can make a complex, they can promote as an activator, they can block as a repressor of the RNA polymerase enzyme. That enzyme is required for transcription of genetic information from DNA to RNA. The important feature of the transcription factor that they contain at least one DNA binding domain. This is very important, that there must be a DNA binding domain, and when this domain is there, if you remember our the last year pharmacology, for each drug we had two things very popularly, that is we call it as a one is for binding with the receptor and another is for the making the action, okay, which is having intrinsic activity. So there are two important domains. So antagonists were not having this another domain, intrinsic activity, so they were just able to bind. So similarly here transcription factor, they must contain at least one domain that is known as a DNA binding domain, and that will cause attachment of a specific sequence of DNA adjacent to the genes that they wish to regulate, or they are responsible for the regulation. They are grouped in the classes based on their DNA binding domains. So on the basis, if you remember, we have seen classification of histones example, okay, and we have seen how they can be classified. The basic 4, or rather basic 5 units. 4 are the core DNA, core histones, and one was a linker histone, if you remember. So all these histones we have grouped into different on the basis of the how they are, what kind of attachments they have, and on that basis we have got that there are many such agents are there. Just a minute, hold on please. Hello. हाँ बोला ना. We were discussing, that is there are transcription factors, and on the basis of their DNA binding domain we have got their classification. There are other proteins also, like they are coactivator, then chromatin remodelers. Remodeler means which changes the shape or structure of chromatin. We have already seen histone transferases and histone deacetylases. We have seen just before this what are the function of histone acetyl transferases and histone deacetylases. Then there are a variety of kinases are there, methylases are there, and they are also essential for the gene regulation. We have seen that how acetylation and deacetylation play important role in the gene expression, how because of that when acetyl is transferred, then because of that how the binding changes, or the bond energy rather we call, the bond energy changes, and because of that the binding becomes different. So this is all what we have studied earlier. So these are all proteins are there, or all these enzymes, these proteins enzymes are there, which are playing important role in the gene regulations, but they don't have the DNA binding domains and they are not transferred. So what is the difference? Although these coactivators, chromatin remodelers, histone acetyl transferase, histone deacetylases, kinases, methylases, although they play important role in a gene expression that we have seen, but they are not the transcription factors. Why they are not transcription factor? Because they don't have DNA binding domain. So this is very important about it. So they are important in the medicine because they modulate, they can cause specific diseases, and we can have certain drugs which can be used to target them, either activators or the antagonist of these transcription factors, and because of that this can be modulated, can be regulated, and we can have variety of actions. So as I told you, the Human Genome Project shows around 1600 these transcription factors, but these are as per human genome. Actually, as per human genome, there are many proteins are there, that is 2800 approximately, but we do not know exactly, sorry, these are having DNA binding domain, I am sorry, this 2800 proteins, they have DNA binding domains, and out of that 1600 are thought to be, they are believed to be having function as a transcription factor, but we don't have exact information of each of it. Okay, as the science will progress, maybe we'll understand still details. So when the next batches will come, when the next batch they will come after maybe few years, 5 years, 10 years, they will have still bigger syllabus than you, because by that time still more information is available and that has been put into it. So these are 1600 are believed to having a transcription factor role, and only 10% of the genes in the genome code for the transcription factor which are making the family. It is the single largest family of human proteins, that is transcription factor. And the genes which are often having the several binding sites, having distinct transcription factor and efficient expression of each, genes that require cooperative action of several different transcription factor. Example, in case of hepatocyte nuclear factors, we need multiple transcription factors. So the combination use, that is 1 plus 1 or 1 plus 2 or 1 plus 3, this type of combinations are there. It is making a subset of approximately 2000 human transcription factor, which are accounting for the unique regulation of each gene in the human genome during the development. Particularly, if you know or if you remember, during development multiple activities are going on, particularly the cell differentiation. This word many a time I have used and I am repeating it, that from a single cell, that is zygote which has formed, which was first diploid cell from 2 haploid cells. So when this diploid cell is formed, from that all human body is formed, which is having more than 300 trillion cells, and in these 300 trillion cells again we have several varieties, and these all varieties having many functions, many different functions, and because of that this transcription factor, and this is only happening because of this formation of different kind of proteins or different kind of material in the cell. Because after all, cell is a functional unit, that is structural and functional unit, and that structural and functional unit will depend only on what kind of structure it has got. Again here protein synthesis play important role, because majority of portion is the proteins, and similarly functional portion, again most of the functions are done by the proteins, because most of the job are done by enzymes, and all enzymes are the proteins. So therefore all these things, they will totally depend on the transcription factors, and thus they are more during the developmental phase. So these transcription factors, they will bind either to enhancer or promoter regions of the DNA which is adjacent to the genes that are regulated by these transcription factors, and depending on the transcription factor, the transcription of the adjacent gene is either up or down regulated. So we have seen that up regulator means increase in the expression, down regulation means decrease in the expression. So decrease, it can be decreased in two ways. Either when we say word this up regulation or down regulation, or promoter or suppressor or repressor. So this word when we use, that means that either two things are happening, either rate is changed. Suppose in previous in 1 second 10 it was there, or 10 genes were done. Now if it is 6, then it is still suppression, okay, or repression, because rate when it was doing 10, now it is doing 6, or it is totally stoppage, it is also same thing. So these are having similar things, that is two important things are there. Anything can be down regulation and vice versa, where anything which can causes from 10 to 15 if the rate is increased, okay, or the gene expression which was not happening earlier, now they are making it to happen. In both cases it is up regulation. What they are doing is, what are the mechanisms by which these transcription factors they are doing their job? They stabilize or block the binding of RNA polymerase to DNA. Stabilize means when it is a promoter one, and when it is the repressor one, or when it is suppressor, they will block the binding. So it is important, because until and unless DNA is bound to RNA polymerase, so this RNA polymerase, until and unless it binds to DNA, it will not able to process it as a for the transcription. So if it stabilizes, then it can do the job in better way. If it doesn't allow or it affects the stabilization, then it will cause the repression. Then it catalyzes the acetylation or deacetylation of histone protein. We know about what acetylation and deacetylation can do just in earlier class. It can either do this directly or it can recruit other proteins which will do this catalytic activity. Many transcription factors use one or other of two opposing mechanisms to regulate transcription. So, these are the important one, as histone acetyl transferase activity. So, we know that is it causes acetylation of histone protein, it will weaken the association of DNA with histone, and that will become DNA more accessible for transcription, and thus it causes upregulation. While histone deacetylation are there, they will cause deacetylation, that will cause more bonding of the DNA with the histone, and the DNA will be less accessible to the transcription, and thus down to the. Now, important thing is how it can be done. Again, here it can be done by two ways. Histone acetyl transposition activity can be increased or blocked. Okay, if it is increased, then the activity, okay, and it can be blocked. So, if it is blocked, means it is something like second activity. Same thing is the histone deacetylase activity, it can be increased or it can be blocked. So, by virtue of these two enzymes, the activity can be done in a four ways. If anything which causes promotion of acetylation, that causes increase in the expression. Those which are causing the decrease in the or affecting it or antagonizing this histone acetyl transperases or blocking it, then they are causing what? The opposite action, that is they are blocking the expression. And if something is increasing deacetylase, they are blocking it. Those which are opposing or blocking it, they are causing increase in the expression. Then in addition to that, this transcription factor can recruit co-activators or co-repressor proteins, so that the transcription can increase. Now, what are the function of these transcription factors? As we have seen, they are important groups, and they are the important groups of the protein that read and interpret the genetic blueprint in the DNA. So, what DNA is meant for? What is the next stages for the DNA? Blueprint means what? Everything is there and all plan of the actions are there. So, it has got that information that what is the next step has to be done, whether this gene is to be expressed or not, or the gene expression which is going on, whether it should be increased or it should be blocked or it should be stopped, or there should be decrease or increase in the rate. So, these all things are there, they will able to sense, they will able to judge it, and there should be just like earlier you have studied in other system, example in cardiovascular system or endocrine system. So, we have got a regulatory mechanism. So, this regulatory mechanism will give the feedback, and similarly accordingly they will able to sense it. On that basis of feedback, they will able to judge that what kind of activities will be there. They will bind to the DNA, they will help and initiate the program of increase or decrease gene transcription. They are vital for the many important cellular process and some of the important function which we can see like that. So, they are important for the basal transcription regulation. Okay. Then these basic one, they are also called as a general transcription factor, that is GTFs, and they are necessary for transcription to occur. Okay. So, they are actually part of the, most of these are the part of the large transcription pre-initiation complex. Pre-initiation means before start, this complex will be there and that will cause interaction with the RNA polymerases. Then what next job they are having is they are having differential enhancement of the transcription. So, differential means one set of the gene will be enhanced while other set will not be having any action, or maybe other set will be having less activity. So, other transcription factor differentially regulate the expression of various gene by binding to enhancer region of the DNA which are adjacent to the regulated genes. And these transcription factor are critical to make sure that the genes are expressed in the right cell at the right time and in the right amount, depending on the changing requirements of the organism. Then next is the development. Many transcription factors, they are in a multicellular organism, they are involved in the development, they are responding to stimuli, and these transcription factors, they are turned on and off the transcription. On and off, who will do? Those which are stimulant, those which are promoter, they will make it on, and those which are repressor, those which are suppressor, they will turn it off. So accordingly, transcription of the appropriate gene will be either turn on, turn off, or rate will be increase or decrease. And then it will change in the cell morphology, or it will activate needed for the cell fate determination. Example, they may synthesize the proteins or the chemicals which are responsible for the cell apoptosis. So the cell will automatically go for the death. Example, RBCs is programmed to die in 120 days. So the fate is known to you. Okay. Maybe in all cells we don't know the fate. RBC is an example. So we know that fate of the cell. So that has been fixed in their, this one. So at 120 days, the proteins will be there, and they will cause the cells to be taken up by the liver, and they will be metabolized. Then they were also important for the cell differentiation. So there are a variety of such families are there. Example of this is Hox family, and there are many such families are there which are responsible for proper body pattern formation in organism. Why we have got a particular body shape. So these are the transcription factors, they are responsible. Then next thing is there is a responsible for the intracellular signal. I told you about, I have given you the example now. What serves as a regulation. So just like we have seen feedback mechanisms, positive, negative feedback in hormones, or you have seen baroreceptor, you have seen chemoreceptor. So these are the signals. They are giving you the signal. They are carrying the signal to a particular place which is supposed to control. So similarly, the cells can communicate with each other by releasing molecules that producing signaling cascades within the another receptive cells, and if the signal requires upregulation or downregulation of genes, they often do it. So example of this is estrogen signaling, that is example of fairly short signaling cascade that involves estrogen receptor transcription factor. It is secreted by the tissues like ovaries and the placenta. It crosses the cell membrane of the recipient cell and bound by the estrogen receptor in the cell cytoplasm, and that will then go to the cell's nucleus and bind to the DNA binding sites, and it will change the transcription regulation of the associated genes. So this is how these activities are done. Then they will also make the response to environment. This is again very important, because just imagine somebody is going to high altitude, there is a lack of oxygen, there is a too much chill environment. So you must be knowing body tries to adapt to the situation in the environment. Now in that case, when this body is trying to adapt to it, then definitely body must have to change or modify the normal physiological mechanism or normal homeostatic mechanism. So in that case, these changes are there. In many times various enzymes are there. These all enzymes on various proteins are formed. So these all proteins, they has to be formed, and they will form only when we know how the proteins are formed. Proteins will only form when there is a transcription. So there are such things are there. Example, they will cause the formation of signaling cascades which are related to biological stimuli. They will also make it example of this is heat shock factors are there, or heat shock proteins are there, HSF or HSPs are there. They will upregulate the genes which are necessary for the survival at higher temperature. Then there are hypoxia inducible factor. I told you at higher altitude oxygen level decreases. So HIF will be upregulate the genes which are necessary for cell survival in the low oxygen environment. Then sterol regulatory element binding proteins, they will help into maintain proper lipid levels in the cells. So this is on the basis of the environment, how it will be there. Then they play important role in the cell cycle control. They are playing role as a proto-oncogenes or tumor suppressors, and they regulate the cell cycle, and they determine how large the cell will be and how it can divide into two daughter cells. Example of the MYC oncogene, which has important role in cell growth and apoptosis. So these are the cell cycle control. We have seen about cell cycle earlier, and this cell cycle is, if it is not controlled, then we have seen that it goes to the cancer. Then they are important in pathogenesis of various diseases. There are many pathogenic conditions are there because of the transcription factor. Example of these are the due to transcription activator like effectors. They are secreted by xanthomonas bacteria. When they are injected into plants, these proteins can enter the nucleus of the plant cells. They will bind the plant promoter sequences. They will activate the transcription of plant genes, and that will help in the bacterial infection, that will improve the, or that will increase the bacterial infection. So this bacteria will decrease the immunity of the plant to have the action on that. So they will contain a repeat region in which there is a simple relationship between the identity of two critical residues in a two sequential repeats and sequential DNA in this TAL effectors target sites. So these are such cases where they are involved in pathogenesis. We shall be seeing about their role in pathogenesis in a human being. So their regulation means, we have seen there are various regulatory mechanisms are there. There are multiple layers of regulations are there, and not only do the transcription factor, they control the rate of transcription to regulate the amount of gene product, that is RNA and protein, but also they are making their own regulation, because they if they are not regulated, then means they themselves regulate their own amount or level or activities. So what are the activities, how these transcription factors can be regulated? So their synthesis can be regulated. There can be, just like feedback, I told you many times today, there are positive and negative feedbacks can be there. So such feedbacks can be there, or sensors will be there, biosensor which will sense the need, and accordingly these transcription factors can be synthesized. Now they are synthesized means what? Again they need to be transcribed. That is from DNA. This DNA will convert into RNA. This RNA will be synthesizing the protein, and these proteins are the transcription factors. So this will be the transcription factor which will be formed, synthesized. Then next thing is their localization in the nucleus. So those which are produced or formed, synthesized, they are transcribed in the nucleus. See, normal other important proteins or other proteins are basically not synthesized in the nucleus. They are actually synthesized in ribosomes or rough endoplasmic reticulum. But here these are the transcribed in the nucleus, and they are then translated into the cell cytoplasm. Many proteins are active in the nucleus. They contain nuclear localization signals, and that will direct them to the nucleus. But most of the transcription factor, the key point is there in their regulation, whether they will stimulate, they will remain in the nucleus, or they are outside. The important classes of transcription factor, like some nuclear receptors, they must first bind to the ligand while they are in the cytoplasm before they can relocate to the nucleus. So they are suppose they are synthesized in the cytoplasm by rough endoplasmic reticulum or ribosomes themselves, then some drug will be there, some agent or some biochemical will be there, they will act on that, and because of this binding they will come to nucleus, and if they come to the nucleus, then only they will able to do the job of transcription factor. How they are activated? This transcription factor must be activated or deactivated. Just like if you remember, last year you have studied protein kinases enzymes. So we have seen that when G protein is activated, it will cause either cyclic AMP stimulation. Because of this cyclic AMP, protein kinases will be there, and you know that this protein kinases will be metabolized by the phosphodiesterases. So we have got two important things. We have seen that protein kinases are the important moieties, and these protein kinases are actually responsible for the basic mechanism of action for what the drug is there or for what that biochemical is working. Similarly, here the transcription factor can be activated or deactivated through their cell signaling domain by a number of mechanisms. One is ligand binding, that is some ligand will be bind to it, and then there are example of these are nuclear receptors are there, NF kappa B, etc. Then it can be phosphorylated, that is many transcription factor like STAT proteins, they must be phosphorylated before they can bind to DNA. Phosphorylation is one of the very important step in most of the activations at many places. Then interaction with the other transcription factors like homo or heterodimerization or co-regulatory proteins, they will be there, and then this will be activated. So this is what actually, let me tell you why everything whatsoever you are studying is. The thing is actually it is a multiple regulatory mechanism, so that at every point there will be check. So for synthesis there will be check, for activation there will be check, for DNA binding site it will be check. So at multiple level check will be there, so that at wrong time wrong gene will not be processed or it will not be expressed, okay, and the wrong amount of the proteins will not be synthesized, less or more. So all these things, that is the schedule, that is it should not be at a wrong time. When it is required it is not happening, and when it was not required it is happening, this is a wrong time. So this is not or this is rarely possible because of this multiple control which are there. Then second thing is their activities, the rate of the activities, it should not be too much, it should be the normal one, and again this is done because of this all multiple regulatory activity. Now how they will be accessing the DNA binding sites? They will be this DNA we know is organized with the histones. We know that this compact thing is called as a nucleosomes, around 147 DNA base pairs, we have already seen that. So these transcription factors, because of this nucleosome, DNA is not accessible whenever it is there, because it has been wound around it. Suppose the thread which is there in the middle, okay, that has been wrapped with the earlier thread, and if some important information is there in the middle, then it has to be, everything has to be uncoiled. So this important thing is that, so some transcription factor like pioneer factors, they are able to bind their DNA binding site on the nucleosomal DNA, but in other cases it should be actively unwound by the molecular motors like a chromatin remodulars or histone acetylations. So because of that, when it is there, when it is unwound is there, then only a specific part will be exposed, and then it can be acting with the transcription factor. So there are two ways. Some transcription factor can even reach to that site of their expression without unwounding or uncoiling of the DNA from the nucleosome, or in other cases most of these will require the uncoiling of the DNA. Then the next is they need to compute for the binding to its DNA binding site with other transcription factor and histone or non-histone chromatin protein. The pairs of transcription factor and other gene can play antagonistic role, that is activators or repressors in the regulation of the same gene. So we can have multiple control. If you remember again ANS, we have seen alpha and beta are having opposite action, or parasympathetic, sympathetic are having opposite action. So this is what multiple control the body is having, so that there will be perfect balance in all this activity will be there. So similarly here also the perfect balance will be there. The other cofactors or other transcription factor will also be there, they will be competing, and accordingly there can be increase or decrease in the activities, as we have seen. Okay, there can be cofactors. Cofactor means which are not transcription factor because they don't have DNA binding domain, okay, but they can increase or decrease the ability of this transcription factor to bind with the DNA. So these are various cofactors, and they will allow the efficient recruitment of these transcription factors. So for a single transcription factor to initiate transcription, all these other protein must also be present, okay, then only it will be working, then only it will be doing the job. and the transcription factor must be in a state where it can bind them if necessary. The cofactors are the protein that will modulate the effect of the transcription factor. They are interchangeable between specific gene promoters and the protein complex that will occupy the promoter DNA. The amino acid sequence of the cofactor that will determine the spatial, here SPTIAL, spatial rather it is pronounced, that is in the space what kind of organization is there, that is called as a spatial. So in the space what conformation is there, that is three dimensionally we have to see it. So in three dimensionally where it is there, if you have studied in chemistry or heterocyclic chemistry or stereochemistry, sorry, that is how the atoms are organized. So this is called as a spatial conformation. Okay, so this is or those if you feel it difficult, please read medicinal chemistry book and see the acetylcholine, because acetylcholine you have studied many a times. You have studied it in APHE, you have studied it in pharmacology, and then you can see that the conformation which it is acquiring, and therefore because of that conformation it can act on different receptor in different way. So this is how that is what the spatial conformation is. Okay. So there are certain steroid receptors, they can exchange cofactors with that is NF kappa B, that is nuclear factor kappa B, which is a switch between inflammation and the cellular differentiation, and the steroids can affect the inflammatory response and function of the certain tissue. So this is why this is one of the important line for you, that is it is one of the mechanism of action of the steroids which are acting as anti-inflammatory. Which steroids are acting as anti-inflammatory? If you remember, we have studied them last year also. At two places we have seen non-steroidal anti-inflammatory. So which are the steroidal one is one thing, and secondary we studied various inflammatory processes. Particularly we have seen bronchial asthma and other such cases wherein we have used certain agents, steroidal agents, which are used as anti-inflammatory. So we have seen anti-inflammatory mechanism at many places. So these are the basic one of the important mechanism by which they are acting as a anti-inflammatory agents. This year also we have studied about anti-inflammatory or immunosuppressant agents. So this is same mechanism they are sharing. The transcription factor and the methylated cytosines in the DNA both are having major role in the regulation of the gene expression. This methylation of various sites of the promoter gene that is usually express the gene transcription, and the methylation is very important. We have seen it how the methylation plays important role. There are around DNA binding site will have around 519 transcription factor. They are evaluated. Out of that, 169 did not have this CpG dinucleotide in their binding site, which is one of the important, and 33 transcription factors they could bind to this agent. So basically what we mean to say is there are multiple DNA binding site and many of the transcription factors are there. Out of that, these transcription factors are acting at one place and may not be acting at other place. At some places they will require the complex formation with other cofactors, as we have seen earlier, and then only they will be able to act. And now structurally all these are proteins. These are having one DNA binding domain, then there will be one activation domain. I repeat, I have make it as simile, or it has matched with the one thing particularly, that is DNA. If you remember last year's pharmacology, basic pharmacology, that each ligand they will have two important sites. One is affinity or binding, and another one is pharmacophore, or which will able to do the action. Okay. So one is similarly here. One will be the DNA binding domain, or the one domain will be binding and other will be the activation domain. And because of this two domain, if they are there, then only it will be acting. Apart from that, it will be one more domain, it can be there, it may or may not be there, that is called as a signal sensitive domain. That is, it will, just like we have told you that there are baroreceptor, chemoreceptor, and they will sense the signal. So this signal sensing is important. So in some cases there will be signal sensing domain which will sense the signal, and accordingly either increase or decrease in the activity can be there. Now we will not go into more details, structural details about each and every domain, but see, on that basis of that there are so many groups are there, though so many families are available, then there are so many response elements are present. That is, because of that they will cause the changes. Lastly, now we are reaching to the important that in various diseases they are found to be involved. If there is a change in the structure or any change in the activity or change in the secretion, increase or decrease, they will be resulting into various kind of problems. So let us see, these are the there. They are very, we have seen they are very important in the transcription or gene expression. So many diseases have been associated with this transcription factor. It may be because of mutation or any other reason it may be there. They are either tumor suppression or oncogene. So we know tumor suppressions, that means they will decrease the cell development, they will increase the cell development, they will decrease the cell growth, and they can increase the apoptosis or cell death. Okay, so they will doing, or oncogenes are there which are responsible for cancers. So the mutation of these, they will result into cancers. There are three groups of transcription factor are known in human cancer. They are NF-kappa B and AP1 families, then STAT family, and the steroid receptors. So they are known to be involved in various kind of cancers. So these are the various diseases like Rett syndrome, diabetes, developmental verbal dyspraxia, autoimmune diseases. There are many autoimmune diseases are there. If you remember, last year you have studied few, this year we can study few. So there are many such diseases are there. Then next is Eli-Frommeni syndrome, breast cancer. Then there are many cancers are there, osteoarthritis. So there are many such are there which are because of the transcription factors. And then we can have various targets for that. Many drugs are there which are working that on that. Example of this is tamoxifen. I hope you have studied these drugs which are acting on estrogen receptor. So tamoxifen is known as a estrogen receptor modulator. If you remember, I don't know, last year pharmacology somewhere you might have studied, it is called as SERM, that is selective estrogen receptor modulator. So it modulates the tamoxifen. Then there is other drug is bicalutamide. It is for the treatment of the breast. Then the transcription factor are often directly modulated by the drug through the signaling cascade, and it might be possible to direct the other transcription factor like NF-kappa B, etc. So there are many drugs now are in the pipeline or believe that they will be there, and thus it can be used, these drugs can be used. Therefore we are studying this. Okay. Now we will not go into more details. We have seen that they play important role in the evolution, because we have seen that example adaptation we have seen like hypoxia induced or heat shock like that. So then if such situation is there, those which are staying in the hot environment, for them it will be permanently, they will have more heat shock like, and their body will be resistant. So this is what we call as a evolution. We will not go into more detail. Okay. These are the classification. Again, these classes are not important, but you just remember that zinc and all these are basically very commonly used. Zinc finger, why it is called as, because around zinc the proteins are having a finger like structure, and therefore it is called as a zinc finger. Okay. So they are classified on the basis of two important things. They are general or upstream one. General means they can form the pre-initiation complex and can do for many activity. They are not very specific for one or other kind of that. But the upstream are there, they are specific factors because they will be doing only for a specific sequences, and they will be depending on the recognition sequences which are present in the gene. So these are variety of such agents which are upstream transcription factors. So I will not go into all these details. These are so many classes are there, functional classes are there. You can remember this, what are the functional basic things are there. Example is constitutively active means they are always present. Conditionally, if you remember last year you have studied constitutive prostaglandin synthesis, which is always there. It is having physiological function. Okay, that is COX inhibitors, they causes even its inhibition. Another one was the selective one, COX inhibitors used, if you remember. So it was, we call it as a inducible, so like that, or conditionally active. So in certain activation is required, that is therefore we call it as inducible. So similarly it is a constitutively active. They are present in the all cells at all the times, while those which are conditionally, they will require certain activation at certain when it will be activated, then only they will be formed. So okay, so some will be only active during developmental stages, some will be signal dependent, etc. So these are very important classes are there, and structurally there are too many classes. I will not go into each and every class how their structures are there, depending on the proteins and depending on the structure they are having. So there are variety of structures are present. Now here we finish all these transcription factors. They are, as I told you, they are important, there are many, but only we are going for the things what their basic studies, and we are not going into the more details. So here we finish the transcription factor. Please go through if you have any difficulties, and please make the note, because if it is there in sessional it will be asked. I am seeing that your attendance is gradually decreasing, but definitely it will be asked because everything is taught, and anything which is taught it can be asked. Okay, just 13 people are there, or 12 people are, sorry, 11 people are there today. So this is very small attendance. So take care and be safe. If you have any difficulties, you can ask. जाधवजी राठोड आहेत का? कुठे बाहेर गेलेत का? बरं बरं बरं, मग आले की त्यांना सांगा मला हे करा. का तिथे? बरं, त्यांनी मला फोन केला तर या म्हणून सही करायला. काय? हां, अच्छा, बरं. हं, बरं, 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 बरं. ठीक आहे ना, ठीक. तुम्ही बरोबर आहे, बरोबर आहे. नाही चालतं. ठीक आहे ना, मग तुम्ही म्हणा मला त्यांचं आलं ना, तुम्ही लेक्चर घेते पण चालते. प्रॉब्लेम नको, बरोबर आहे, बरोबर चालते ना. हं? ओके, चालतं चालतं. तुम्ही सांगाल तेव्हा आपण करून टाकू. ओके सर, थँक्यू. फार्मॅकोलॉजी पण सेकंड इयरला. काय झालं? हम्म. हम्म. अच्छा. हम्म. हम्म. सांगून द्या ना, असंच सांगितलं असं सांगितलं. यांनी सांगितलं नाही ना काही. एक टॉपिक मला ते डाऊटच यायला लागला, म्हटलं आणि प्रेमात एक टॉपिक मागे. बस संपलं. मग फार्मॅकोलॉजीला माझ्या हात लावायचा नाही, तर गोंधळ करेल, सगळं गोंधळ येतो. माझ्याकडे फार्मॅकोलॉजी पहिल्या दिवसापासून आहे. त्याने आज पहिला टॉपिक घेतला एक समजा, तर त्याला हां, ते तर नशिबाला, त्या एवढं पुण्य कोणाचंच नाही. मी येतो खाली 5 मिनिटात आलोच नाही.